Is Bath & Body Works Worth the Money? By Lexi Conda, Sydney Gortel, Julia Kelly, and Elena Swingen. For our research project, we asked the question, does the price of hand sanitizer affect how effective the product is in killing germs? Our purpose was to determine whether the generic or Bath & Body Works scented hand sanitizer was more effective in killing bacteria. We hypothesized that the Bath & Body Works hand sanitizer would be more effective than the generic hand sanitizer in killing microbial growth due to it being more expensive. Before applying hand sanitizer, we took a control sample from the index finger and streaked it into the control portion of the agar plate. We then applied the generic hand sanitizer to a single hand and swabbed the remaining 3 fingers. Each swab was streaked into 3 separate quadrants. After swabbing one hand, we switched, obtaining a control from that hand and streaking the plate into one quadrant. Next, we applied the Bath & Body Works scented hand sanitizer. We gathered a sample from the remaining 3 fingers and streaked those into 3 separate zones on the agar plate. After all of the samples were gathered, they were incubated for 48 hours. After those 48 hours, we observed the growth and number of colonies.
In the data we collected for our control, there was an average of 390 bacterial colonies growing per plate sample before the use of hand sanitizer. The data we collected after the use of hand sanitizer determined that generic hand sanitizer still had an average of 1 bacterial colony per plate with a standard deviation of 1.46. The Bath & Body Works hand sanitizer reported to still have an average of 1.28 bacterial colonies per plate with a standard deviation of 3.04. This overall showed an average difference of 0.28 between the 2 hand sanitizers. By running a paired t-test between the 2 experimental groups, we calculated a p-value of 0.29. This value is greater than the alpha statistic of 0.05, which leads to a rejection of the null hypothesis. After conducting a t-test for the control, a p-value of 0.001 was determined for both Bath & Body Works and the generic hand sanitizer. Since the p-value is smaller than the significant value, the null hypothesis is accepted that the control is different from the experimental groups. These results contribute to the field by observing the effectiveness of antibacterial solutions in killing bacteria living on our hands.
So here we've included some images of the results that we obtained just so that our process could be better understood. Take note that for each petri dish, quadrant 1 was our control. Quadrant 2 through 4 were all added into our experimental data points. In order to quantify our results, we used a few forms of statistical analysis. First, we decided to do some bar graphs with standard deviation error bars, and then we conducted a t-test to determine p-values. So standard deviation error bars quantify the scatter among the values. Looking at whether or not the error bars overlap lets you compare the difference between the mean with the amount of scatter within the groups. When looking at figure 1, it appears that there is no overlap between the error bars of the control and the error bars of both of the experimental groups. This indicates that the difference is likely very significant. When looking at figure 2, however, the error bars for the 2 different experimental groups of Bath & Body Works and generic hand sanitizer, they have quite a bit of overlap, indicating that they are likely not significantly different. Bar plots and error bars do not allow you to be sure that the interpretation is correct, though, so you must conduct a t-test that takes the sample size into consideration. Next, we conducted a paired t-test, otherwise known as a two-way t-test, in order to compare the average between the generic hand sanitizer and Bath & Body Works hand sanitizer. Because our p-value was 0.29 and it was greater than the 0.05 level of significance, we failed to reject our null hypothesis and concluded that the generic hand sanitizer and Bath & Body Works hand sanitizer are not statistically different. The p-value for the comparison of Bath & Body Works with the control was 0.001, and the p-value for the comparison of the generic hand sanitizer was also 0.001. Because both p-values are smaller than the 0.05 level of significance, we accepted the null hypothesis and concluded that the 2 experimental groups are significantly different from the control.
Moving on to the discussion section. Due to not enough evidence, we cannot make any conclusions to support the hypothesis that Bath & Body Works hand sanitizer is more effective than generic hand sanitizer in killing microbial growth. In order to properly determine which hand sanitizer was more effective, we performed an experiment that would independently measure bacterial growth on nutrient agar plates. We selected a sample size of 6 people. With each subject, we tested 4 fingers, one of those fingers being the control. Therefore, with 6 subjects testing the effectiveness of hand sanitizer on 3 fingers, we came up with 18 different samples. Each individual tested was given 2 separate agar plates. One plate was designated for Bath & Body hand sanitizer effectiveness, and the other plate was designated for generic hand sanitizer effectiveness. In order to keep our results fair and without bias, half of the plates for each sanitizer were performed on the left hand and half of the plates were performed on the right hand. In order to use aseptic techniques and decrease the risk for cross-contamination, each individual wore a sterile plastic glove on the hand not being tested. To start the procedure, each person labeled the 2 agar plates, one named Bath & Body and one named generic. We then had each individual divide their 2 nutrient agar plates into fourths. Zone 1 on the plate was where the control finger, the pointer finger, which was kept free from any hand sanitizer, was swabbed. The next 3 zones were introduced to the hand sanitizer. Zone 2 on the agar plate tested the middle finger with hand sanitizer. Zone 3 on the agar plate tested the ring finger with hand sanitizer, and respectively, Zone 4 on the agar plate tested the pinky finger with hand sanitizer. Once the control finger, the pointer finger, that was not introduced to hand sanitizer was swabbed with a Q-tip and smeared on the agar plate, the other fingers could come into contact with hand sanitizer. Each finger respectively was swabbed with a Q-tip and smeared on the agar plate. We incubated the plates for 48 hours so that enough microorganisms would grow to show results, but not too many that we would have our results be too many to count. As you can see from Figure 1, titled Average Comparison with Control, introducing hand sanitizer had a significant effect when compared to the control group, the dirty finger. This makes sense as each hand sanitizer has 99% effective written on the bottle. Figure 2, titled Average Comparison Between Experimental Groups, did not show any significant difference. The error bars on the 2 experimental groups had overlap. This confirmed that neither one nor the other is more effective in killing microbial growth. The statistical analysis paired t-test compared the 2 hand sanitizers. With a p-value of 0.29, it indicated that there was no true significant difference in the effectiveness of killing microbial growth in Bath & Body hand sanitizer and generic hand sanitizer. The statistical analysis t-test showed the p-value of 0.001, indicating that the 2 experimental groups are significantly different from the control. Therefore, based on our bar graphs, charts, statistical analysis, we concluded that there is no significant difference between the 2 hand sanitizers due to not enough evidence. However, we were able to draw the conclusion that both hand sanitizers were effective in killing microbial growth. It was interesting to compare the differences in prices. Bath & Body Works hand sanitizer averaged $1.75 per ounce, while the cost of generic hand sanitizer averaged 45 cents per ounce. Because our results showed no significant difference in the effectiveness of killing bacterial growth in the 2 sanitizers, Bath & Body Works has no evident benefit for the higher price. We did encounter some problems throughout the experiment. There was a wide range in bacteria growth in the control group. Some subjects had a control of too many to count, while other subjects had a control of only 1. Because the control number varied so greatly, it affected the other results when calculating the mean. Each subject could have been exposed to different bacteria throughout the day. Each subject may have also sanitized their hands more or less frequently than the others. Additionally, the swabs could have been contaminated when transferring to the agar plate. The amount of hand sanitizer could have varied per subject, as some may have put more or less on their hands than others. For improvement in this experiment in the near future, we would recommend methods to reduce contamination and uncertainty in results. We would recommend that the hand sanitizer is measured out in specific dosage amounts to reduce the risk of skewed results. We would also recommend having the same individual swab each subject. Our technique of acquiring the sample was not consistent, as we had some subjects swab themselves and other subjects swab each other's fingers. This may have skewed our results, as some subjects may have swabbed more surface area or swabbed longer than others. Finally, in conclusion, in the relation to the academic community, our group was not able to find any research that was similar enough to ours to draw a comparison from.
In conclusion, we reject our original hypothesis that Bath & Body Works hand sanitizer is more effective at killing microbial growth because it is more expensive. By comparing the averages on a bar graph, we are able to determine that due to the overlap in error bars between the two experimental groups, there is no significant difference between the two. The p-value calculated from the paired t-test determined that there is not enough evidence to support the null hypothesis. There is not a significant enough difference to say that one hand sanitizer is more effective than the other. The cost of the product did not seem to change how effective the product was in killing bacteria. However, we concluded that there is a significant difference between the control and the experimental groups based off our p-value calculated from the t-test for control. This concluded that hand sanitizer is overall effective in killing bacteria.